Hello, everyone. Welcome to today's live broadcast, USP 232 and 233, Understanding Method Requirements and Guidance for Getting Your Laboratory Ready for Implementation. I'm Laura Bush, the Editorial Director of Spectroscopy, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by Spectroscopy and sponsored by Milestone. Over 20,000 customers worldwide use Milestone's products for microwave acid digestion and solvent extraction, synthesis, and ashing, as well as acid purification systems and direct mercury analyzers for solid, liquid, and gas samples. Their consultative partnership approach and the trust of their customers have led to Milestone's becoming an industry expert in a wide variety of applications, involving some of the most challenging matrices. Milestone's mission is to enable laboratories worldwide with success, growth, and continuous innovation. I'd also like to mention our special guest today, Chemical Solutions Limited, or CSL. CSL is an independent testing laboratory specializing in trace-level elemental analysis. Established in 1995 and providing testing using ICP-MS since 1997, they are solely committed to the analysis of metals, minerals, and other elements. Their dedication to their customers has quickly established them as an expert on USP 232 and USP 233 implementation. We have a few housekeeping announcements before we begin. The webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, which you can find on the right-hand side of your screen. You can enlarge the slide window by clicking on the small icon in the upper right-hand corner of your window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the web seminar. If you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your window. And please also note the resources made available from our sponsor, which you can find in the green resource widget below your window. I would now like to introduce today's speakers. We're very pleased to be joined today by Francine Walker of CSL and Johan Nortia of Milestone. Francine Walker has over 25 years of laboratory quality control and manufacturing experience. She has implemented and administered quality systems for a variety of laboratory and manufacturing facilities to meet the requirements of ISO 9001 and 17025 and the FDA's Good Laboratory Practice Guidelines. With extensive experience in the analysis of metals using a variety of instrumental and wet chemistry techniques, Francine is considered to be an expert in the field. As the technical director of CSL, Francine frequently implements testing innovations. Johan Nortia received his BS in chemistry from the Southwest Baptist University in 2005 and began work as an applications chemist for Milestone in 2006. Johan's primary focus and responsibility is to educate and train Milestone's customers on microwave digestion and to develop applications for preparing samples prior to ICP-OES and ICP-MS analysis. Francine and Johan, thank you both for joining us today. Francine, you have the floor.
Thank you, and good morning. I'm very pleased to be able to discuss this very interesting topic with all of you today. I'm just going to do a brief introduction to Chemical Solutions to tell you about the history that we're going to be bringing into our presentation today. As Laura had mentioned, Chemical Solutions has been dedicated to only testing of metals, trace metals, minerals for about 20 years now. We do have 5 ICP-MS units and 2 OES units that are dedicated to that type of testing. I'm not going to spend a lot of time going into our background, but that history does bring in capabilities and knowledge from a variety of different matrices and different products. We work in the pharmaceutical industry as well as with excipients, active ingredients, foods, medical devices. So a lot of what we bring to the table and some of the data that we're presenting today will be knowledge of internal standards and testing techniques that have evolved from that history and out of those particular capabilities. Some of the accreditations that we bring to the table but is not required in the USP testing arena, we do have an FDA registered and audited facility. NELAC brings an environmental background focus that we have had from our historical perspective. then ISO accreditations, and a lot of memberships that have helped us over the years in the learning that we're going to bring to the table today. So let's take a look at the regulations and some of the regulatory efforts that we're going to discuss. Obviously, the seminar focuses around the general chapters 232 and 233 of the USP, those being the elemental impurities limits, the limits and the elements of interest as defined in the chapter 232, but also 233, which is the procedures the actual methodology that allows us to reach those limits and those levels as defined in 232. Also, part of the USP discussion, or at least in the elemental impurities arena, is the historical 231 chapter, which we'll discuss very briefly, and the 2232, which is specific to dietary supplements and the elemental contaminants as such. Now, the ICH has certainly been in discussion with regard to the USP for quite a long time. So we're going to discuss the history there, a little bit about the background and how the two, USP and ICH, have been in the process of aligning their elemental impurities limits and processes. The EMEA, the European Agency, the European Medicines Agency, and the European Pharmacopoeia are big players, as well as many U.S. companies are not unique to the U.S., and they are obviously shipping products or registering products in a variety of countries. So we're going to be discussing the European procedures and their limits as well. So let's get up to speed. Let's start by looking back quickly at where we've come from in the arena of elemental impurities. The general chapter in the USP 231 has been titled Heavy Metals, and it's been around for roughly 100 years. Most people don't understand that although it's one general chapter, it actually encompasses 3 different methods. There's a method 1, 2, and 3. And those methods are specific to the type of materials that were being put through that particular process. By definition, what registers as positive in the 231 or any of the metallic impurities that can be colored by a sulfide ion. The general chapter has called out 10 individual elements. You can see that in the second bullet point there. Lead, bismuth, arsenic, antimony, tin, some of those elements that are represented there were thought to be responding to the sulfide ion. A lot of data since that time, since that chapter was written, has suggested that those all really aren't very responsive to that particular methodology. 231 is very difficult, whether you're doing the method 1, 2, or 3 portion of that general chapter, very difficult to conduct. It's been shown that mercury really does not react very positively, so it can be missed very commonly in that particular type of test. It's also very limited in its sensitivity. It's a test where you're looking for a 10 ppm plus or minus. So you're looking at a 10 ppm limit, so you're looking to see whether you're below that limit or above that limit. And if you look at this slide, you can see the picture there, or actually 4 samples that a customer of ours had transferred into us that they had been receiving 231 type numbers for a number of years as either positive or negative above that 10 ppm. And you can see this being a color test. The left-hand tube is actually the lead standard at 10 ppm. And with the color that appears in those particular solutions, it's very difficult, if not impossible, to say whether or not those particular materials were above the 10 ppm limit. And the importance of this is just that a lot of people come to us and they're trying to translate their historical testing data with regard to heavy metals to their new data, which is being generated under the 233 methods. And there's very little comparison between the two. So unfortunately, most historical data generated by 231 is not going to be particularly relevant for comparison's sake to the newer testing limits and methodologies. The ICH, let's talk a little bit about this ICH group. The ICH, just the acronym, stands for the International Conference on Harmonization. It was launched in 1990, and it was a group of people from the regulatory authorities and from pharmaceutical industries in the European Union, Japan, and the U.S. So it started with those three countries. More recently, Health Canada and Swissmedic have been much more instrumental in that group. The intent here, and it's a very good intent for anybody in the pharmaceutical industry, is to provide some harmonization on the interpretation and application of the technical guidelines and requirements. So for those of you working in the pharmaceutical arena and trying to transition products in and out of various countries, it's very cumbersome. So this group really was trying to get the groups together, harmonize, come up with uniform standards such that testing performed to meet one application would then be transferable fairly readily to another application. I'm going to kind of go through this a little bit quickly, but elemental impurities, the Q3D is a quality directive for coming out of the ICH, and it has been adopted pretty substantially across the globe. The European Union, Japan, Swissmedic, and the FDA now are all on board with what have become known as the Q3D guidelines. So let's take a look at the 232. So the ICH has been working on guidelines, and now the USP has also subsequently been working on guidelines as well. The 232 is thankfully now in its most recent draft form as of March 1st, 2016, now in alignment with the ICH. Previous versions have not been in such good alignment. The 233, the general chapter that talks about the testing methodology, that has been unchanged for quite a while now, but this 232 general chapter has been moving. The takeaway from this slide might be that application set date of January 1st, 2018. That's when the general chapters 232 and 2232 will be in place, and the 231 general chapter will be removed from the monographs and from the compendia. On a closer look, this is again a bit of the history of the elements and their limits as shown in the 232 general chapter. You can see in 2013 we had, and in early 2015 as current, we have a lesser group of elements than are included in the ICH classifications and in that ICH elemental impurities arena. You can see that the limits have moved as well. Just looking at arsenic, we can see in 2013 we had a regulatory level of 1.5 called out. That has now moved upwards to 15, whereas cadmium had been at 25, and that has moved down. And on this slide, I'm specifically showing the oral permissible daily exposure level that is called out in 233, and that's based on a microgram per day limit. So the per day refers to the serving size, the daily serving allowance for any particular material. We're going to talk about that more as we go through the slides here. You can see on the left-hand portion, the ICH classifications, those come out of the ICH document. Class 1 is the highest priority, sometimes called the Big Four: arsenic, cadmium, lead, and mercury. Those are elements of particular interest because of their toxicity as well as their nature as being more prevalent in materials found in pharmaceutical materials. Another quick look at the ICH as compared to the USP. We can now see very quickly that the elements and the levels are in alignment between the two agencies. And we can see that there also from this slide that the limits are based for both of these on the route of administration. So you'll see different levels for each element, whether it's an inhalation sort of hazard versus if the material is used in a parenteral fashion or administered orally or ingested as part of its usage. And you can see that you must know the element and the route of administration to determine what your target levels are going to be if you're testing for compliance to the 232 general chapter. Now, part of the 232 that is very undefined is the idea of how much testing you have to do. Certainly, the chapter requires a risk assessment to be done on your materials. And the risk assessment, if you look at the general chapter 232, defines what needs to be included in that assessment. Again, this doesn't define testing needs, but rather just the assessment of whether or not it should be included in your risk analysis. So certainly any element from that list that has been intentionally added as part of the processing must be included in the thought process for risk assessment. If not intentionally added, you then go to the route of administration again, and you're looking to see whether or not that needs to be considered in your risk assessment. The Class 1 or the Big 4 elements, arsenic, cadmium, lead, and mercury, certainly have to be considered at all times, as do Class 2A. Then going below that, Class 2B and Class 3 of lesser toxicity and lesser likelihood of being present. Whether or not they need to be included depends upon the element and the route of administration. Okay, so now that we have some background and we know what we're testing to, let's talk about some of the choices we have to make to come into compliance. There's a bit of a puzzle that has to be solved when you're looking to comply with the 232 general chapters of USP. There's choices to be made as to how you're going to look at your products, whether they're going to be raw materials or finished products, what instrumentation you're going to use, how you're going to prepare the sample, and how you're going to analyze those particular materials. So looking for a systematic approach to doing this, going from top to bottom is your most broad perspective thoughts, and then your choices are going to narrow as you go down through your process. But from a compliance perspective, at the top level, you have to decide whether or not you're going to comply by testing or reviewing, doing a risk assessment of your finished products or your raw materials that go into those finished products. Choices have to be made on what elements you're going to include in your risk assessment, and that's partially addressed in that previous table, the limits you need to hit, and whether or not you're going to do some sort of pre-screening of these materials or go into a validation of a particular testing method. Preparation, as we're going to see through a broad piece of our conversation today, is an extremely important part of the process, and certainly one that is not well defined in the general chapter 233 for the methodology itself. We're going to talk a little bit about the choices that could be made there as to whether or not you're going to work on total impurities or allow the option or accept the option of bioavailable impurity methodologies for preparation. And then we're going to talk about some instrumentation choices as well. Broadly, the ICP MS versus OES as called out in the 233 general chapter. But there are a lot of details, again, that are not called out. Choices to be made in terms of how you tune that instrument, what modes of operation you may select to use, calibration levels, etc. So very broadly, in any finished drug product, you have not only your active ingredients, but you have other pieces of the puzzle or other processes that you have to think about. You have excipients, you have water that may be used in the processing, you have the containers and closures that your material may be packaged in, you have the equipment that it touches along the way. Any of those pieces may be a potential introduction of an elemental impurity. So if you're looking for a compliance option to do your finished product, you have encompassed all of those potential contamination sources into one. Otherwise, you may have to at least think about each of those pieces independently. So again, talking about the compliance, there's two options, generally speaking. You can approach the compliance from your finished product perspective, in which case you're looking, again, at all of those individual pieces, your containers, closures, all the things that touch that product in one piece. And there's certainly some value to doing that. First of all, the limits in the microgram per day level that they're defined in 232 is very much more applicable to a finished product because you know a daily serving size on a finished product where you wouldn't if you were trying to use a raw material. So raw materials excipient providers have a tougher time complying to this particular general chapter because there may not be one defined serving size for their material. So that makes the testing a little more challenging in defining what the specification for a raw material may be. Another thing to point out here on this slide is this idea of a J value. J is just the term used for specification value in 232. And that's very important because you have to know what specification you're targeting to fully comply with the 233 general chapter testing methodologies. So as you decide how you're going to test either finished product or raw materials, you now know what you're going to test, but how do you do it? And here are some more choices that we have to make. In the general chapter 233, which is the methodology, you have the option of a neat, a direct aqueous, direct organic, or an inorganic preparation. An inorganic or indirect preparation is one that requires heating of the material. That automatically talks to the idea of a closed vessel digestion where you're avoiding the loss of any particular volatile element that or component that might be lost in a preparation piece. So I know many of the people on this call or potentially going to be on this call had some ashing equipment that they had utilized for testing metals in the past. Because of the high heat involved in ashing and the open nature, that would not be a particular technique that would fit well within the definition of 233. The other thing with 233, we talk about a visual clarity. The preparation at the end should be fully dissolved, or the material should be fully dissolved. So that has become known in the industry as visual clarity. So you only know something's fully dissolved if you visually cannot see it any longer in solution. From an analytical perspective, you have choices to be made on what instrument that you use. There are some alternatives possible, but typically they're talking about MS or OES, ICP-based methodologies, and whether or not you are going to perform screening, as we said, more of a screening, or use a validated technique. Again, calling out the specific targets of J being that specification. And then you're going to do calibrations based upon that particular spec and then running standards at half and twice that particular spec. And let me try to talk to this a little bit more in depth. What we see here is a theoretical calibration for curve for cadmium. In this particular case, this is just representing the idea of defining your specification for a particular element. You have to define it for testing purposes in parts per million or part per billion rather than in micrograms per day. So the thought process here is that the cadmium, if it were to be called a specification of 8 ppm, which would equate to the current ICH guideline of 5 microgram per day for an average size tablet, which is about 0.6, 0.7 grams. So in assigning your calibration for cadmium, you have to know a couple of things. One, you need to know the specification. Two, you have to take into account the initial sample, starting sample mass that you're going to use for digestion, the final volume of that digestion, and what subsequent dilutions you may need to get that particular material to behave well on your instrument and in your calibration range. One of the tips that we recommend to people who contact us, if they're setting up a calibration curve, is to set your standards in multiples of 2. That way, since you're trying to hit a specification and half of that spec and twice that spec, you have the ability to change that secondary dilution factor and still allow it to hit your particular curve. So in this case, the J value, if I did a 1 to 5 subsequent dilution on my initial digest state, would equate to 16 ppb, and I would have 2 points above, one above and one below that. But if I had to dilute further, it would fall further down on my curve, but I would still be within the guidelines of half J and 2J. Now, there's a lot of complicating issues once you get to the instrument. Again, nothing that's well defined in the method. Choice of internal standards can be very critical. Whether or not you have unfiltered particles, if you don't have a complete digestion, we have seen very complicating issues relative to that. Your choice of acids for the digestion can be critical as well. If you need to use hydrofluoric acid for that digestion, you can see that in some cases that would be counterproductive if you were to be trying to test for calcium or magnesium with that same digestion fluid. So something, one of the reagents that may help to dissolve one material may actually cause a precipitation of another. And this is some real data, just very briefly. You can see across the bottom different internal standards that lead can be tied to by ICP-MS. This was a real live material. It was a cementite clay that had some of these naturally occurring elements in. And they were introduced in a mixed fashion, so they are all introduced at the same level. But by making a different choice in the internal standard, you can get very differing values for lead in the exact same digestion. I'm going to show some quick data with regard to sample preparation, kind of leading into the topic of sample prep. We had done some work on cementite clay, and we did 6 different varying digestions and aggression. We had done some shaker methods, which were they were not microwave-based. This was for some bioavailability work that we had done for a particular client, where we changed the extraction fluid from the weakest of a DI water to the strongest in that shaker method of a 1 normal nitric acid. And in between, we had a 0.5 normal hydrochloric and some weaker hydrochloric. Then we moved to microwave methods with a final temperature of 175 degrees versus 37 for the shaker. And we changed the concentration and the aggressiveness of the digestion. I'm just going to show you briefly how that might affect the outcome. What you see here again is the cementite clay. So the elements at the bottom are actually the main constituent elements of the clay. So they're found in very high concentrations. From left to right, you see a gradient of low to high concentration that's very, very specific to the aggression of the digestion. So if you're using a very weak preparation technique, you can get levels 10-fold, 20-fold, 1,000-fold below that of a more aggressive digestion. And what's relevant here is aluminum and magnesium follow that pattern. A more aggressive digestion brings out more of those particular elements in your final digest state, whereas sodium and calcium, we did not see that. So those are very easily removed from that particular clay material. So the digestion wasn't quite as important for them in this particular case, but it was certainly very important for some of the other elements. I'm also going to add in that in adding back that water component, you see an anomalous, what appears to be an anomalous blip in the aluminum result, but that is relative to unfiltered particles because this was not a full dissolution. And these are the impurity or the lower level constituents of that cementite clay. We will see here that, again, the aggressiveness of the particular digestion plays into how much comes out. You can see the nice sloping results for the increasing intensity of the digestion, except for lead, which we had called out before. The internal standards tied to lead could make that value of that right-hand bar go up or down quite dramatically depending upon choices made in the analyticals there. Okay, I think I'm going to turn this over to Laura. Laura, I think you have a polling question for all of us.
Yes, I do. Thank you very much, Francine. For all of you in the audience, we'd appreciate it if you can participate in our poll, and all you have to do to participate is just to click directly on your screen to answer. So here's the question. What have you found to be the most challenging aspect of getting your laboratory ready for the USP changes? Changes of implementation date, shifting of method requirements, evaluating instrumentation to purchase, or training and getting operational on purchased instrumentation? Once more, you can click directly on your screen to answer. What have you found to be the most challenging aspect of getting your laboratory ready for implementing the new USP chapters? Changing in the implementation date, changes, shifting of the method requirements, evaluating instrumentation to purchase, or training and getting operational on purchased instrumentation. All right, let's pull up those results. So we've got kind of a range of answers here. Looks like about even amounts of you said that a challenge was shifting of the method requirements, and also an even amount said training and getting operational on purchased instrumentation. But clearly there's a bunch of you concerned about the changes in implementation date and evaluating instrumentation to purchase. Excellent feedback. All right, well, let's continue with the presentation. Johan, over to you now.
Thanks, Laura. So jumping off of where Francine left us, her last couple of slides there really indicated how critical thorough sample prep is to the trace analysis process. And while sample prep can sound simple, as Francine mentioned, there are certainly a number of variables that one should keep in mind both when doing the preparation but also when evaluating different systems to do the preparation in. Milestone has been in the sample preparation business for close to 30 years now. We have about 20,000 units installed globally, and many of those units are being used to do pharmaceutical digestions, whether it be raw products, excipients, final products like tablets and capsules. So we have definitely experience with these types of matrices, and if you look at method 233, what you'll find is that the method is fairly nondescript in terms of telling you exactly what to do. Certainly there are some broad categories to talk in terms of neat, indirect, and so forth. For today's discussion, I'm going to focus on indirect, which, as Francine mentioned, really implies microwave digestion. Some specific parameters that we have seen to cater to these types of samples. Firstly, in terms of sample size, it really does vary depending on the type of testing that you're going to be doing. As Francine mentioned, whether you're doing your final products like tablets and capsules, your sample sizes might be a little larger. And if you're doing raw products, perhaps your sample sizes will be a little bit smaller. So there is a bit of variation in terms of the sample sizes that typically get processed. In terms of an acid chemistry, we found that for most labs, a combination of nitric acid and HCl will do the trick. And certainly if you have some siliceous material or need to look at silica, then you'd have to add HF to get a complete digestion. Temperature is a very critical component of the sample preparation process. And we've really found that there is a large sort of variance between the different temperatures required to get different samples into solutions. We've done work anywhere from 200 to about 250 degrees Celsius to get some of the more challenging samples into solution. So when laboratories are evaluating sample preparation equipment, it can be overwhelming dealing with all the different variables that are out there. We like to focus on a few key questions before recommending a certain system. And the most critical of that is, of course, the sample types that you're going to be processing. As I mentioned, depending on whether you're doing raw or final products, you may need to adjust the temperatures and subsequent pressures that the samples will require to go completely into solution. Additionally, we need to understand the elements of interest, and of course that's prescribed in the methods. So fairly clear, as that gives us an indication of the acid chemistry that's going to be required. To indicate how critical temperature is to the digestion process, I wanted to share a slide here in which we're digesting a fairly challenging matrix. It's a polymer, and you can see on the left-hand side, we've digested that polymer at about 180 degrees Celsius. And when Fran mentioned earlier and spoke about visual clarity, you can see that that sample doesn't have any visual clarity. You have a lot of flocculants, a lot of particulates remaining, and that really becomes challenging when you go to the post-digestion step and the analytical step. On the right-hand side, we've taken that exact same sample and we've increased the temperature about 40 degrees. And you can see that we've now achieved that visual clarity that we're looking for. But an important parameter that I wanted to have you notice is the fact that the pressure that was generated in that temperature bump and in thoroughly digesting the matrix was about threefold. So we went from about 20 bar of pressure being generated to about 60 bar of pressure being generated. So when you're looking at systems, it's very critical that the system can handle both the temperature but also the subsequent pressure that the digestion will generate. When you look at the market today, you'll find that systems predominantly fall in one of three categories. The first is sequential, the second is rotor-based technology, and the third is single reaction chamber. And I'm going to take a few minutes to talk to you about each type of technology and their pros and their cons. First off is sequential technology. Now, this technology was born in the synthetic laboratory, and as the name implies, you're running samples one by one. In the synthetic environment where a lot of work is done around R&D, this is ideal because you're testing different solvent systems, different temperature, different catalysts. So running something and knowing what the result is advantageous. Ultimately, though, because you're running samples sequentially, it becomes challenging to scale that as throughput needs increase, which a lot of the USP chapters are written really around a QC function, and so throughput should increase as you're testing more types of samples. The other downside that the sequential systems have is that they can be fairly limited in temperature and pressure, simply because a lot of synthetic work isn't as aggressive as doing a digestion. Next up, I'm going to spend a little bit of time talking about rotor-based technology. Now, I know many of you on today's webcast have some experience with rotor-based technology, so I won't take too much time. But essentially, in a rotor-based system, you have a rotor that is comprised of a number of individually pressurized segments. The nice thing about these rotor-based systems is that depending on the types of samples that you have, you can choose different rotors to meet your needs. In our new Ethos UP, which is Milestone's rotor-based system, we have two types of rotors. The first is a high throughput rotor. It's called the Maxi 44, which enables you to process up to 44 routine samples per batch. The other rotor is called the SK15. That's our high pressure, high temperature rotor. And really what we've found is that a lot of the labs getting ready for the USP implementation have gravitated towards this rotor, as it enables you to get to higher temperatures, higher pressures, and subsequently ensures a more thorough digestion. Another thing that we've really focused on when we designed the Ethos UP was around simplicity of method development. As Francine mentioned, when you get into sample prep, there are a few variables that are going to impact the quality of your digestion. And certainly, temperature is one of those. What we've done in the Ethos UP, the system comes pre-programmed with 300 digestion methods. So once you log in, you'll simply go to the icon-driven menu, select the group of applications that you'd like to run, in this case pharmaceutical. From that menu, you'll select a matrix that matches what you'd like to run, and the system isn't only going to know which time and temperature to program, but it'll also give you, the operator, some guidance in terms of the acid chemistry as well as sample size that's required to achieve a thorough digestion. When the system then runs, you'll have a live real-time view of the temperature profile to ensure that you do in fact get to your final temperature for a thorough digestion. The Ethos UP has been extremely popular, and rotor-based technology continues to be what most people associate with microwave sample prep. That being said, rotor-based technology does have some limitations. In this rotor-based technology, you typically have a reference vessel, and that vessel is being continuously monitored for temperature, and subsequently the microwave is adjusting microwave output to ensure that our desired temperature is maintained. That results, though, in the need to batch different types of samples and different chemistries together. So what you'll find is you need to run your raw products, your final products, and so forth. So there's an element of batching required. Additionally, because the rotors are polymer-based, there are some limitations in terms of the temperatures and pressures they can hold, and certainly for some of the more challenging matrices, that could become a limitation. Additionally, there is some handling required, assembly and disassembly of vessels before and after the digestion, and they may also require cleaning from one run to the next. So about 10 years ago, Milestone set out to find a way to get around some of these challenges that rotors present. And we ended up designing a technology called single reaction chamber, or SRC technology. And it's become extremely popular. We have more than 600 systems now globally, and it's been particularly popular in the markets of petrochemical, pharmaceutical, and contract testing. And the fundamental difference that single reaction chamber has is that instead of working in individually pressurized reactors, as you would in a sequential system or in a rotor-based system, in single reaction chamber, you work in a single stainless steel reactor. Within that reactor, you can place up to 22 vials or different digestions, and those vials sit in a water bath, which we call the base load, in the very bottom of the chamber. That base load and subsequently the samples that sit inside of them ultimately get monitored throughout the process to ensure that we reach the correct temperature to have a thorough digestion. It's a very key concept that all the samples within the chamber are being processed under the same conditions, and therefore we can really ensure that reproducibility is maximized in this type of setup. So how does single reaction chamber really work? Here's a bit of a flow sequence in terms of how you'd run it in the laboratory. Sample gets loaded into a vial. We have a couple different vial choices. There is a quartz vial, a Teflon vial, and a disposable vial. And those vials get placed into a rack, which gets lowered into the sample chamber. Prior to starting the run, the chamber is pre-pressurized with nitrogen. And in a second, I'll tell you why that's such an important step. The microwave program then commences. At the end of the run, there's an automatic depressurizing step. So instead of having to depressurize individual vessels like you would in a more traditional setup here, all the depressurizing is happening automatically by the instrument. The rack is then hoisted out of the chamber, and you can simply dilute it and take it to the ICP for analysis. A common question that we get is how in the system, since you don't have a complex assembly of vessels, we ensure that we don't have any cross-contamination. As I mentioned, prior to starting the microwave program, we will pre-pressurize the chamber with nitrogen gas. And it's an essential part of the process, as that pre-pressurizing ensures that the boiling point of all the acids within the chamber is immediately raised. And therefore, there's no cross-contamination between samples since you don't have any boiling or splattering of the samples. And to indicate that, we ended up doing a run here of 15 vials in which some of the vials were loaded with about 110 ppm of mercury. So as you know, mercury is very volatile, has this tendency to move around. And at the end of the run, we analyzed the blank samples to see whether there was any carryover. And as you can see from the data there, they came back with no mercury in them. So we're very confident that even for very volatile elements like arsenic, like mercury, you're not going to have any cross-contamination. So really this concept of a single reaction chamber has fundamentally shifted the way that laboratories do sample preparation day in and day out. In the traditional rotor-based setup, because samples need to be batched, what you'd essentially have is in each rotor, you have a sample type. So you'd have to do one run doing your tablets, and that will take you about an hour or maybe an hour and a half to load samples, unload, and so forth. And then on your next run, you'd have to do your raw powders and then your excipients. And ultimately, the time for each one of those microwaves run get compounded. So it can become a fairly long process, and you're really not maximizing the productivity of the instrumentation. The real shift that single reaction chamber has enabled is because we are measuring the temperature of the chamber and not of any of the individual samples, we can group all the different sample matrices together prior to the microwave run. So now, instead of having to do a rotor which is only partially filled, we can combine our different samples, whether it be a raw product, a final product, or an excipient. And even if the chemistry is different, we can put them all in the same sample rack and run them in the ultrawave at the same time. So you really have a way of getting a quality digestion, reproducible, but also really maximizing the productivity around the instrument. To give you an indication of how single reaction chamber stacks up to even a high throughput rotor, we wanted to see how many samples we could process during an 8-hour day. And at the end of the day, we found that running a single reaction chamber, even though you're running 15 samples at a time compared to 40, you're actually able to process 30% more samples in a single reaction chamber system. And that doesn't even count the fact that in a single reaction chamber system, you could run much more challenging samples and get higher quality digestions because you can go to such high temperatures. We found that the ultrawave and single reaction chamber technology has been particularly popular in the pharmaceutical laboratories for a few different reasons. Firstly, because you can go to such high temperatures and pressures because of the stainless steel construction, you don't have to guess about the quality of digestion and the temperatures required to get a thorough digestion. You can simply pick a very aggressive program, and regardless of the sample matrix that you're running, you will get a good digestion. Because we don't have a reference vessel like we would in the rotor-based approach, we can combine our different sample matrices. So that really causes an efficiency in the laboratory to maximize throughput. Because there's no assembly, disassembly, and depressurizing of individual vessels, labs find that the UltraWave is a very easy system to use and easy for operators to get trained and comfortable with. And last but not least, we found that the UltraWave over a period of time has substantially lower consumables cost. It's about a quarter of the cost of some of the traditional rotors in these rotor-based setups. So with that, I'll give it back to Laura for another poll question.
Thank you very much, Johan. For all of you in the audience, we do have another question for you, and once again, click directly on your screen to answer. So here's the question. In complying with the USP general chapters, do you anticipate needs in the areas of method development and or validation? Yes or no? So once again, in complying with the USP general chapters on 232 and 233, do you anticipate needs in the areas of method development and or validation? Yes or no? We'll just give you a quick moment there, and then let's pull up our results. So the vast majority of you, just about 85%, have said yes, which I guess is no surprise. There's a lot to be done. Okay, well, let's continue with our presentation. So now back to Francine to wrap up.
Thank you, Laura. Well, with regard to method validation, certainly that is a piece of the puzzle as we're analogizing the implementation to a puzzle that presents some challenges in and of itself. In a general sense, method validation has been around as addressed by the USP in General Chapter 1225 for quite a bit of time. And the important part of this is the fact that they are testing to be by laboratory studies and meet the requirements for the intended analytical application. So validations in a general sense typically address some of these particular elements. They generally address all of these elements, but to a differing degree. And again, with the compliance to the General Chapter 232, we're looking at an appropriate amount of compliance. So obviously accuracy, precision, specificity, LOD, LOQ, your quantification, your detection levels, all of those pieces of the puzzle are important in any validation. In the respect to complying with the 232, some of them have more importance potentially than others. Obviously, accuracy and precision are important no matter what you're looking at. Specificity is actually listed on this slide twice, and actually that is a component that I find often is overlooked in the validation protocols that people are developing. We're going to go into some example protocols that comply with 233, but may be missing that specificity piece. LOD, LOQ may not be quite as critical because now you're looking to target testing around a particular specification. Some of those elemental specifications are quite high, so trying to tweak a method or a validation to show that you can detect chromium at sub-ppb levels really would not be time well spent when you're talking about compliance levels up around 11,000 ppb, or excuse me, ppm in some cases. So looking at an example validation protocol, and there are many ways to approach this, and pharmaceutical protocols can be quite lengthy, but in its simplistic sense, you're looking at the quality objectives, which we just discussed. You're looking for accuracy, precision, intermediate precision. You're making some choices as to what type of measurement activities you're going to assess to comply with those objectives. Most often, we talk about running control samples or calibration standards controls in deionized water at a variety of levels, representing the specification for the element of interest, and half and either twice that specification, or more commonly 1.5 times that particular specification. So you're running multiple levels of standards. You're then going to assess your test material or your test sample, which is your material of interest, spiked at each of 3 different levels, again, referencing right around that specification that you've determined for that particular element and for that particular serving size. You also are going to have to measure unspiked samples, obviously, to assess the recovery of the accuracy spikes that you are doing. From a precision standpoint, we typically look at 6 spiked samples and compare the numbers generated for each of those, the recovery numbers, and look at the relative standard deviation. How precise, how much can we replicate the recovery of those spiked samples? Intermediate precision, ruggedness, there's a lot of variability on what people are looking to assess here. Oftentimes, it's changing up the variables in terms of a different analyst to perform the testing, a second day for the testing, if available, a second instrument for the testing. The more variables you change, the more in tune you are with the ruggedness or the reproducibility, if you would, of those particular numbers. Specificity in terms of false negative and positive. In terms of the 233, really cue in on the accuracy requirement. So the thought is if you're recovering your spikes as you're performing for your accuracy, you're showing that the methodology is specific for that element in that material. And what we've seen over time is that's really not always the best thought process, because if you have something that falsely looks like at your instrument level, say mercury, tungsten can interfere with mercury. So if you're looking at a dietary supplement which has a significant amount of tungsten in it, tungsten oxide, if it's oxidized, and it commonly does become oxidized in an ICP-MS unit, will look like mercury. And that false background is there, and you can recover, you can get good spike recoveries on top of that, but really not be assessing the unspiked material that you're looking at is truly mercury. In this particular case, we had a client who tied the specificity to accuracy. We generally try to shy away from that and to specify a different technique, maybe ICP-OES if the level's high enough, a different detection technique, or at least monitoring a second isotope on the MS if that's a valid option. So bringing in, again, the idea of the puzzle. You started the process with making decisions on whether you're going to use raw materials or your finished product. You've put it through a digestion process. You've put it onto the instrument, and you've generated some data. You've decided on a validation protocol to see if that data is good. So let's take a look at some of the results that were generated on a particular material that we just ran. Going back to the validation protocol, which we just talked about, these are some of the results that were achieved. Now, these were achieved using an ICP-MS. This was done on a Nexion instrument. It's a Perkin-Elmer unit. This was done with varying elements being in varying modes. We have some elements, as you will see here, that we ran in standard mode, others that were run with the KED or the kinetic energy dispersion mode turned on to eliminate some of the background interferences. You can see that in this particular validation protocol at the end, we defined an element, we defined an isotope for a particular element, the internal standard that was tied to that element to produce the end data. So at the end, you have a very robust method. You're sure that that particular isotope, that particular internal standard, and the recoveries that you specified in the protocol are being met. And you can see here that there are a variety of elements done at a variety of specification levels. And this is an abbreviated table just for the purpose of being able to fit it on one slide of some of that data that was recovered. And in this particular case, you will see the highest recovery for this spiked material for arsenic. And if you look closely at the validation protocols as defined in 233, you will see that there's an allowable recovery for spiked materials up to 150%, and that was very purposely revolved around the arsenic spike recoveries, which typically do run above 100%, and that has to do with the concentration in the background, a very heavy matrix, very ionic in nature. To get to those low levels tend to make your arsenic recoveries higher. And there are some techniques to bring that down, but the 150% recovery limit for that particular 233 general chapter really was because of the arsenic. Looking at the second analyst, same idea. In this particular case, the protocol called for a second analyst to run 6 spike samples only at the specification, not at multiple levels. And again, using the particular ISA and isotopes for the elements, putting the sample through the ultrawave digestion unit, which was used in the ICP-MS, we saw excellent recoveries for that validation. The FDA does have a lot to say about validation, and these are some quotes from a website that monitors the FDA compliance. They've had a lot of observations recently regarding validation verification. Many people think because you're using a compendial test method that it's a vetted method and there's no need for verification or validation. And certainly we've seen a lot of 483s issued that are publicly available that speak otherwise, that they are looking to see some sort of testing to be done to show that the method is representative or appropriate for the sample that you're using. And I'd just like to end the presentation today before turning it back to Laura with a favorite quote of mine: quality is certainly never an accident. It's the result of intelligent effort and a lot of intelligent choices which are to be made if we're going to come into compliance with the 232 and 233 general chapters. And over to you, Laura.
Great. Thanks so much, Francine and John, for your excellent presentations. It's now time for questions from the audience. Let's just dive right in here and get started asking some of them. Okay, so first one, what are some good standard reference materials that you and your clients use in for example, certified in a number of trace materials?
That's an excellent question and one that people struggle with significantly. Because you're talking about complex matrices, finished materials, no two finished materials are identical to each other. So to find an absolute reference material that matches your sample is often hard. We use a lot of materials from the NIST. They have a wide selection of materials. They are not very, the price points are pretty high, so they're not available at a discounted rate by any stretch. We also use the CRN out of Canada. They do have some materials that are maybe more applicable to certain matrices than are available through the NIST at times.
Excellent. Does this apply to topical use pharmaceuticals? And if so, what is the limit for topical use pharmaceuticals since 232 is based on oral, parenteral, and inhalation products?
Excellent question as well, and one that we hear often. There are a lot of models that have been developed by different organizations that use the oral PDEs as a baseline for the topical specifications. And then there are different exposure modifications or factors that are applied based upon how likely the material is to be absorbed through the skin. So the FDA, while they don't have any direct guidance out on that, many industries have algorithms out there that have looked at their particular materials and how much may be absorbed through the skin. There are some training modules being developed right now by the ICH. They're a working group within that agency which talk to some of those modules, and offline, if anybody would like to have some access to that, I'd be more than happy to get that to them.
Fantastic. What is your recommended method for validating recovery of elemental impurities from the sample? Comparison to a CMR, if available, linear correlation to sample mass, or other?
I'm just going to refer back to a lot of the work that we have done. We have most people wanting to do spiking at multiple levels of the material itself in the specific digest state that's generated during the digestion process. When available, we can take CRMs, certified reference materials through the process, but again, it's very rare that they are identical to the material of interest, so it's very difficult to tie it back to a CRM directly.
Do we expect the new USP 232-233 regulations to apply to cosmetic over-the-counter products?
Yeah, that's a great question. Boy, we've got great questions on the line today. There is no direct reference to cosmetics in the particular general chapters, but because some of the active ingredients in those particular type of materials can be considered active pharmaceutical, I know they are kind of on the fringes of this. At this point, we don't see a lot of activity in the cosmetic industry, in our cosmetic clients in complying or the need to comply with the 232.
Right. And for everyone in the audience, we're just about at the top of the hour, but we'd like to go just a couple minutes past the hour because we have a lot of good questions. So if you can just hang in there with us, we'll try to get a few more questions answered. Question about digestion. Can we digest oil samples also?
Absolutely. So with the ultrawave, again, working in stainless steel, sometimes oils can be challenging in the traditional rotor-based setup. It certainly can be done and depends a little bit on the viscosity and so forth. But between a high-pressure rotor and certainly on the ultrawave, we have a lot of users that are using the system for digestion of oil products. I don't know, Francine may be able to comment a little bit about how they group between going to the rotor-based system and the ultrawave. Perhaps in this case, I would presume that she would probably tend to lean towards the ultrawave for this type of challenging matrix.
Yeah, that's correct. We do run oil samples through both rotor-based and the single reaction chamber, the ultrawave systems that we utilize here. Typically, our go-to is the single reaction chamber, the ultrawave, because of the higher pressures that it can tolerate and the more complete digestions we can get out of that at those higher temperatures. If you need to use a larger volume of oil, however, it can be very aggressive. So the larger vessel containment, the opportunity for those larger vessels in the rotor-based system sometimes do play into the need to use those.
Excellent. Johan, I think this one's for you too. What is the tolerance of similarity in matrices in the Ethos UP? For example, different batches of the same type of pharmaceutical products from different manufacturers.
Yeah, it's a very good question because it's such a gray area question. It's extremely hard to try and quantify what we would consider to be somewhat similar or very different. I think if you try to make it very black and white, really any sample matrix that you presume will either be a little bit more or less reactive or will require a different acid chemistry should really be batched separately. So, there are certain instances where if you're running a sample and it's in the general ballpark, the composition is somewhat similar, you could get away in a rotor-based setup. But really for those laboratories who are running a variety of different matrices and different chemistries for those matrices, a single reaction chamber system is probably the better setup if you're kind of on the fence as to whether samples are or are not alike.
For ICP and ICP-MS, which one of the instruments do you recommend to use for the USP methods between ICP-OES and ICP-MS, I guess is what the person's asking.
We typically use the ICP-MS because of its availability to go to the lower levels that are needed for certain elements. Obviously, given some of the specifications that are there, the OES would hit those levels. It's not quite as sensitive as the MS. In validation efforts, where it's available and where we're able to do so, we will put the particular material on both instruments, kind of that dual detection for the specificity component. But it's always easier to further dilute your sample to the more sensitive instrument. So if you have to use one or the other, the MS makes a lot more sense from its sensitivity standpoint.
Great. For excipients or raw materials, how do you best decide on J as a microgram per gram spec?
Another great question and one we hear often. The raw materials or excipients, we typically direct our customers to look at the usage of those as best they have control of that usage. Oftentimes they're selling them to an end user which may not disclose the actual formulations on their end. But look at the highest usage for that particular material and use that for your specification, your daily limit for specification calculation, because that way it's always safer to be a little below where you want to be. So if the usage is a little less, that is fine, but use your maximum known serving size for that when you set up your validations or your testing.
We manufacture an excipient which uses no metals of concern in its production. We intend to screen at the USP levels for the various metals on representative lots. Do we need to perform the screening for each lot? Is screening adequate as an alternative?
Again, great question. There's no requirement for testing here at all. So in terms of screening, what you have to do is do a risk assessment. And in that risk assessment, if you find enough variability from even the certificate of analysis from your material providers, or if you have done some testing and you see some variability, certainly the thought process would be you'd have to test more frequently, and that can be every lot, it might be every third lot, or every time you change a manufacturer. People are looking at that very closely to try to minimize the amount of testing, but make sure they have covered their bases to assure that they're releasing a good, solid finished product.
Great. Let's just do one or two more because these are good questions coming in. The use of microwave is for solids and hard to dissolve solids. Do you have any situation in which the microwave would be needed for parenteral injectables?
Francine, you want to take that one?
We certainly have not. Oh, I'm sorry, Jan, go ahead.
No, go ahead. I was going to pass it over to you.
Okay. We've had some application with the parenterals if they're very organic in nature and you're testing by ICP-MS and you're using the traditional 52 isotope for chromium. If you haven't eliminated the carbon from the parenteral, you can get into some difficulties with trying to analyze chromium, again at mass 52, because of the carbon content. So in some cases, it's not for visual clarity that you want to perform the digestion, but sometimes just to remove the background constituents.
The highest level recommended in the USP is 1.5J. So why test up to 2J?
Yeah, there's been some back and forth on that particular issue. The methodology calls for, has varied a little bit over the course of the initial iterations. They talk about J and half J and 2J, I guess because of the multiples of 2 being a little simpler to calibrate. The validation now, when they have you spiking, they've more commonly gone to or called out 1.5J, so you're a little closer to that specification. So that's been an area where that has been back and forth a little bit, 1.5 versus 2J. That has changed over time.
All right, let's take one last one. I was informed that new NDA or ANDA submissions need to demonstrate compliance with USP 232 and 233 by June of 2016. Can you confirm that, and do you have any particular suggestions about ensuring compliance?
The June 2016 is a European deadline, so the directive over there has been 2016. So we have seen people needing to be in compliance a little earlier than the 2018. What happens in 2018 is the use of 231 in the actual monographs goes away. So you will no longer be allowed to use that. So to be in compliance, obviously you have to do your risk assessments and any testing that you may want to do up front and include that in the application. Most applications or all applications that we've been involved with, they don't want to see just initial pre-screening. They almost always want to see a little bit of testing and some validation or verification that that testing was appropriate for the particular material of interest.
Fantastic. Well, we need to wrap up now. I thank you both, Francine and Johan, for your talks and for answering all those questions. That was great.
Thank you.
I'd also like to thank all of you in the audience for participating today, and I'd like to thank our sponsor, Milestone, for making today's webcast possible. This webcast will be available for on-demand viewing through May of next year. You'll receive an email from Spectroscopy alerting you when the webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.